In chapter 7, now we're going to look at the acellular or non-living microorganisms. So first of all, just how we describe these, because they are not living. Again, they're not cells, but they usually impact human life and other, and can definitely be pathogens. So instead, the best way to describe viruses are active or inactive instead of alive or dead, because again, none of them are alive. So that gets used interchangeably, especially in just common language. But again, just technically, we talk about viruses as being active or inactive. Again, they're not considered living because they can't multiply independently of the host. They only have a small genetic code, whether it's DNA or RNA, so they're completely dependent on their host to get energy and to reproduce. However, they do have they are able to direct certain processes. So essentially they can go into the cell and essentially take over. So that makes them more than inert, kind of lifeless molecules. So they're in this kind of gray area. So again, the term active or inactive is what we use. Also the term obligate intracellular parasite, because it requires that virus to invade a specific host using its genetic and metabolic machinery to release new viruses. So again, they're specific. We'll talk about host range and organisms that they can or cannot infect. But again, they have to have that host in order to reproduce. Again, they're not just agents, though, of disease. Some of them actually influence the host genetic makeup. So they can be, their viral DNA can be inserted into the host DNA, so it can shape the way that cells and tissues, whether it's bacteria, plants, or animals, have evolved to their present form. So it's estimated that between 40 and 80% of our genome is actually made up from various viral infections, because again, that viral DNA or RNA comes in, and some of it can actually become integrated or part of the host. So we have some kind of descendant viral DNA within our genome. And also they are part of our normal microbiome. And the microbiome are all the living, or all of the organisms, the microscopic organisms that typically reside in or on our body. So part of our gut has lots of different bacteria and microbes. So all of that community is referred to as the microbiome. So some of the properties of viruses, so quite a few here. Again, they're not cells, they're not living. As we already mentioned, they're obligate intracellular parasites, so they have to have another host. But not all of them use humans. There are viruses that infect bacteria we'll talk about, and all other cell types. They are inactive macromolecules outside of the host. Again, they can't survive on their own, but once they infect the cell, they become very active. And the overall kind of basic structure is they have a protein shell that's called a capsid, and that surrounds their nucleic acid. And as we mentioned before, that can be either DNA or RNA, but not both. They're found everywhere in nature and hugely impact life. Ultra microscopic, smaller than bacteria, smaller than any of our cells. Just to kind of put it in perspective, between 20 and 1500 nanometers. Again, not a number you need to know, but just kind of putting it in perspective of how small these viruses can be. As I just mentioned, they have DNA or RNA, never both like cells do. And then that DNA can be double-stranded, it can be single-stranded, same with the RNA. RNA can be single or double-stranded. So in our cells, DNA is double-stranded, RNA is single-stranded, but viruses can have kind of a variety. So carrying the molecules on their surface determine the specificity. So again, viruses are specific either to like a host, so they can either infect like humans or possums or E. coli or staph, but not a virus can infect every living thing. So it is very specific because they bind to receptors on the cell. And sometimes, especially with viruses that infect animals, it's specific to a tissue type. So they can only infect, say, T cells of our immune system, not every single cell in the body. And they multiply by taking, again, control of the host so that the host cell becomes this virus producing factory that makes the new viruses for it. And they usually lack enzymes because they use the host cell, but sometimes the virus will have something specific like HIV. It's an RNA virus that needs to make DNA. So it has a specific enzyme that takes RNA to DNA. Our cells don't have that. So the virus has to bring it with it. And again, they usually lack machinery like ribosomes to make proteins because they use the host cell. All right, so just a kind of basic structure for most viruses. Again, not all viruses are the same. But the most simple structure, again, we have that genetic material. Again, either DNA or RNA, never both. They have this external protein coat that surrounds that. Again, some of them can have some enzymes inside there too, but essentially they have this protein coat. They can have a variety of shapes, and then their nucleic acid, DNA, or RNA inside. Again, maybe some enzymes, and then some of them can also have an envelope on the outside. So we'll talk more about the difference between enveloped and naked coming up. All right, so some of the components, so the protein shell that surrounds it is called a capsid, and then the term nucleocapsid is the capsid plus the nucleic acid. So again, some of them can have an envelope. It's not the same as a cell membrane. Again, even the enveloped are not considered cells. It's usually modified from the host cell membrane, but again, not all viruses have them. So those that lack an envelope are called naked, and those that have it are called envelopes. And then they also have what are called spikes, and these are on both naked and enveloped viruses. These are the projections that stick out from that capsid or the envelope, and it allows them to dock on the host cell. So this is what makes them specific and allows them to attach to the host cell. All right, so that, again, this generalized structure. So the virion is the fully formed virus that can establish a host. Again, it's not a living thing, but this is ready to go, ready to attach and cause an infection. So we have this nucleic acid, we have the protein coat called the capsid, and then these spikes that stick out and allow it to attach. And then some of them may also have this envelope outside of the capsid and that make them enveloped. Now, the way that they reproduce or multiply has several stages to it. So we'll go through each of these, but it's important to know kind of the stages because this is where we can look at targets and how to alter the viral infection. So first they have to bind and be absorbed into the host cell. Again, even inside of the human, they can't just reproduce in the blood. They have to be inside of one of the cells within the organism. So they have to attach, they have to get, again, penetrate or get in, they have to uncoat or remove that capsid to release their DNA or RNA. Then they have to take over the cell and cause the cell to make its proteins and its nucleic acid to replicate it. Then it has to put all that together and then ultimately get released from the host. So if we look at each of these, again, the first step, absorption. So this is that invasion. So it has to encounter something susceptible, some susceptible host. Again, viruses can't attach to every cell. They can't attach to every organism. So it is specific using those spikes. And then it's literally absorbed, again, at a specific receptor site on the cell membrane of the host. So absorbing just means it's attaching to it. So we think about like soaking in a paper towel or something like that. So the attachment of the virus using those spikes to the host cell receptor is the first step. This term then host range, this refers to the different types of organisms that the virus can infect. So it can invade into its host cell. Again, it has to be compatible. So the restricted host range, for example, hepatitis B, only infects liver cells of humans. So if it comes in contact with a muscle cell or an immune cell, it doesn't attach, it doesn't absorb, it can't infect those. It has to be in the liver and only in humans. Hepatitis B doesn't infect raccoons or horses or anything else, only humans. So it has a very limited host range. Then a moderately restrictive host range. So this is, for example, the polio virus. It infects different types. So intestinal and nerve cells of primates. So all primates could be infected by polio. And then one with a broad range, like rabies, infects lots of different cells within the host, but all mammals can be infected. So again, the different number of different species that the virus can infect, or the different number of cell types within a species, determines whether it's restrictive, moderately restrictive, or broad. All right, so the penetration and encoding, this is again how once it attaches, that absorption, then it actually has to get into the cell. Typically endocytosis is used, which is what we see here. So we get the spikes attaching to the receptors. That triggers the host cell to essentially engulf it or eat it. And that's referred to as endocytosis. And now the viral particle is inside of the cell. Now it has to go through this uncoating step. So now we have to remove whatever the host cell membrane is. We have to get rid of the capsid so that that free RNA or DNA can then be inside of the host cell. There's also showing here this irreversible kind of attachment. So again, it attaches here, and instead of going through endocytosis, it just fuses. So an enveloped virus, where again, this is a naked virus, it doesn't have an envelope. Here with the enveloped, instead of using endocytosis, the membrane from the virus and the membrane from the host cell can actually join together, and it just becomes the same membrane, and that releases the capsid into the cell. But it still has to release its DNA or RNA into the cytoplasm, and that step is referred to as uncoating. In the synthesis, this is just producing the genetic material and then any proteins that the virus may need. So that nucleic acid, whether it's DNA or RNA, that cell is going to be directed to make viral DNA or RNA. The DNA will replicate in the cytoplasm. DNA, though, will be in the nucleus, because that's where those typically take place inside the eukaryotic cell. Now, once the virus is actually assembled, the proteins have been made to make up the capsid, any spikes, the nucleic acid has been replicated, that all assembles inside of the cell, and then it actually needs to get out of the cell. So the enveloped viruses, they're going to get released by what's called budding or exocytosis, so the opposite of how they got in. So again, we'll have this assembling happening where this viral DNA is going to come, or RNA. It'll get surrounded by this the capsid proteins, and then essentially it's just pinched off. So endocytosis brings things in, exocytosis takes them out. So it'll essentially pinch off a piece of the host cell membrane and then release the virion that has that envelope. Now then, the naked viruses are more going to just slice the cell. So it would rupture the cell, and that would release the virions. Now, a little bit here about different types of infections from viruses. So persistent infections, what happens here with these is we get a carrier relationship. So instead of the virus coming in, taking over, releasing the virions or lysing the cell, instead it kind of coexists with the host. So the cell harbors the virus, and again, it's not immediately lysed to release them. So now what happens is some of the viral DNA becomes incorporated into the host DNA, and this is referred to as a provirus. And what this does is it creates a chronic latent state where there can be periodic reactivation of the virus. So essentially the viral DNA, instead of replicating, assembling the virus, and releasing, it just kind of hangs out inside of the host cell, and then it can be reactivated. And this is what happens with herpes. So there's the cold sores or the genital herpes, what happens there is that's why it kind of goes into a latent state. So the viral DNA is always going to be part of that host, but there can be periods where it's latent and there's not an active infection, but it can become reactivated later because that viral DNA is still inside of the host. Unfortunately, the incorporation of some viral DNA can also lead to cancer. And these are referred to as oncogenic viruses because they're viruses that can lead to cancer. And it's estimated that about 13% of cancers are actually due to a viral infection or the presence of a virus. What happens here is a process called transformation, in which the virus carries genes that can directly cause cancer. So when they get incorporated into the host genome, it causes those cells to over replicate, leads to tumor formation, and then ultimately cancer. So those viruses can also produce proteins that induce a loss of growth. So either way can lead to a cancer. Either it's the viral genes that cause the replication, or it's the production of various proteins that cause the cell to keep dividing and dividing, even though it shouldn't be. Now, there are also viruses that infect all cells, but we'll talk a little bit about ones that infect bacteria, and they're called bacteriophage or phage or just phages. These were actually discovered in the early 1900s. Most of them are double-stranded DNA, but they can have other genomes. But essentially what happens is they infect bacteria. They parasitize it, they take over the bacterial cell, and then ultimately kill it, and then release the virions as they escape. So often they make the bacteria that they infect more pathogenic. So just like in humans with the latent infections, sometimes the phage DNA actually becomes incorporated into the bacteria that they infected, and that gives them an extra advantage. It gives them extra proteins that they produce, and it makes that bacteria more pathogenic. So they can kind of live, kind of coexist as well. Other times they go in and they lyse the cell to get out. So these bacteriophage, the most common one that's typically shown is one that looks like this. It's one that infects E. coli. It's one of the most well-studied ones that's there, but it goes through the similar stages of an animal virus. So it still has to attach, it has its spikes, it inserts its DNA, causes the cell to replicate the DNA and make any proteins it needs, it assembles, and then it gets out. They can also go through this kind of coexisting, though, too, like a latent infection. It's called lysogeny. So these are specific types of phages called temperate phages. So they can undergo absorption and penetration, but again, they don't immediately go through replication. So instead of a provirus that we talked about with the latent and persistent infections, these are prophages. This is the inactive state where that phage DNA is inserted into the host. And then through a process of what's called induction, it activates the prophage of that cell and causes it to replicate and then go through lysis to get out. So again, just like with animal viruses, they can kind of coexist, either proviruses with animal viruses or a prophage whenever we're talking about bacteriophages. But again, they incorporate their DNA into the host and can be inactive for a time. Now, the danger with this, and the reason we're talking about it as far as with human diseases, is that those phage genes in the bacterial chromosome, whenever it becomes kind of latent or inactive, it can actually cause the bacteria or allow it to produce different toxins or enzymes that increase that pathogenicity. So they make it more pathogenic. And that process is called lysogenic conversion. So this is where the bacteria acquires a new trait that came from one of those phages. So it's actually viral DNA incorporated into the bacteria, but now it produces something that can be harmful. And just some examples, diphtheria toxin, cholera, and botulism, those toxins are all produced because of viral DNA that's been inserted into their genome. Now, when we look at ways of treating viruses, there aren't a lot of targets because viruses, first of all, they mutate very rapidly, so it's hard to design therapies. One of the most common approaches is actually prevention, so developing vaccines that prevent infections. As far though as treating them, there aren't nearly as many antiviral drugs as there are antibiotics because viruses don't have a lot of targets. Again, they're a protein coat and they're nucleic acid. So there is some success if they have special enzymes like HIV, that's a retrovirus, and it has a specific enzyme that our cells don't. Those can be targets. But again, most of them don't have that. So it's really hard to treat them. So vaccination is the most common. But also some success with interferons, which is actually naturally made by our immune system. But there's been some success at preventing and treating viral infections using interferons kind of more synthetically or as a treatment. And then even the bacteriophages that we just talked about, again, they can cause problems with bacteria being more pathogenic, but actually some bacteriophages may be useful in treating some viruses, treating some bacterial infections later on. So we might be able to use them for bacterial infections. But again, as far as treating viruses, it is very tricky because there are so few targets. Now with some of the other non-cellular infectious agents besides the viruses, we'll also talk about the prions. So prions cause what are called spongiform encephalopathies. So spongiform because it makes essentially the brain into a sponge, and encephalopathy is relating to the brain. So this is only like a neurological disease. It doesn't affect any other part of the body, but it is a chronic and persistent disease. It's not treatable. There's no way of altering the prions that cause the disease, so it can't be treated. So the brain tissue removed, though, again, looks like a sponge or just a bunch of holes placed in it. So these prions are that common feature between these, and these are distinct kind of proteins deposited that can affect these animals. So what this actually looks like, so how that infection works is unknown. So again, the protein composition, it doesn't have DNA, it doesn't have a cell, nothing. It's just a protein that is able to essentially infect the brain. It can destroy brain tissue, it can affect other proteins. But again, how exactly it works is still not completely well understood. And again, treatments are not yet available.